Hello everyone and welcome to Big Things Made Simple. Today we will be talking about eukaryotes and prokaryotes. First we will answer the question, what is a eukaryote? We will then go on to talk about prokaryotes and then compare the two. So, what is a eukaryote? These are organisms whose cells store DNA in the form of chromosomes contained in a distinct nucleus and whose organelles are membrane bound. All organisms fall into this category apart from eubacteria and archaea. An example of these are plants, animals and fungi, all of which can be seen on the right. As we said, eukaryotes are defined as having membrane-bound organelles, which allow for what we call compartmentalization. An example of common organelles found in eukaryotes are the nucleus, rough endoplasmic reticulum, ribosomes, the Golgi apparatus, mitochondria, and vesicles and vacuoles. We can look at these organelles and their function in an animal cell. The diagram shows the basic structure of an animal cell. If we start from the left, we can see that there is a double membrane porous nuclear envelope, chromatin, which is loosely coiled DNA wrapped around histone proteins, and the nucleolus, which is the site of RNA synthesis, which together make the nucleus. We also have ribosomes, which are essential for the second stage of protein synthesis. Translation. We have rough endoplasmic reticulum. This is a flattened cisternae system with ribosomes attached to it and is involved in the making, folding and modifying of proteins while providing a pathway to transport materials. We also have smooth endoplasmic reticulum, which is a tubular cisternae system with no ribosomes and is involved in the making, storing and transporting of carbohydrates and lipids. Next, there are vacuoles. In some organisms, these are food vacuoles formed by phagocytosis of food particles, and in others, such as freshwater protoctists, these are contractile vacuoles which pump out excess water. Next, we have mitochondria, with the inner folded membrane of cristae and 70S ribosomes. These are essential for ATP synthesis, respiration, and lipid synthesis, and basically provide the energy for the cell. This is all suspended in the cytoplasm. Cytoplasm provides cell shape and is the site of much cell activity such as metabolic reactions and consists of water, dissolved solutes and other insoluble suspended particles. There is also the Golgi apparatus. This is a complex series of curved membrane cisternae and is involved in the modifying, sorting and packaging of proteins and lipids received from the endoplasmic reticulum. You will also find that there are lysosomes. These are sacs of digestive enzymes that digest unwanted organelles. This is all protected by the plasma or cell membrane, and this is a selectively permeable phospholipid bilayer and provides protection for the cell and controls what enters and leaves the cell. You also have microfilaments made of actin which help to aid cell movement, intermediate filaments which provide mechanical support, centrioles which are 9 sets of microtubule triplets in a ring shape and are essential in cell division in the formation of spindle fibers. And then finally we have microtubules, which provide both cell movement and mechanical support. These together form the cytoskeleton, which is not always defined as an organelle, but is still an essential structure in the cell. Plant cells are also eukaryotes. While they have a very different structure to animal cells, they share many common organelles, as you can see here. However, plant cells have further organelles, which animal cells don't have. These include the cell wall, which is made of cellulose and provides cell shape, strength, and protection because the cellulose fibers are inelastic and thus prevents the cell bursting. Plant cells also have a large central vacuole with a tonoplast membrane filled with fluid containing dissolved solutes and other particles, and its function is to regulate the osmotic properties of the cell and provide support for the cell. Plant cells also contain chloroplasts. These are small, flat, and double-membraned organelles with 70S ribosomes, and is the site of photosynthesis and ATP synthesis, and so is essential to cell function. They also have peroxisomes whose two functions are to 1. Break down fatty acids in order to form membranes and to be a fuel for respiration, and 2. Create H2O2, hydrogen peroxide, to convert into water for the cell. They also have plasmodesmata, which are strands of cytoplasm that run through tiny pores in the cell wall throughout the cell and provide pathways for cell-to-cell communication. Now that we've been through eukaryotes, we can now talk about prokaryotes. Prokaryotes are organisms that store their DNA in the form of a circular loop that lies freely in the cytoplasm and has no membrane-bound organelles. These organisms are usually unicellular and tiny, and a common example of this is bacteria, which you can see an example of on the right. Like we said, prokaryotes have no membrane-bound organelles. An example of these include the nucleoid, which contains DNA in the form of a circular loop lying freely in the cytoplasm, the flagella, which is the tail of the cell and is needed for propulsion and movement of the cell. They also have pili, which are organelles that stick out of the cell and are needed for bacterial conjugation. These can also take form as fimbriae, which are thinner hair-like structures and are needed more so for attachment. They also contain plasmids, which contain genes and are essential in DNA exchange and antibiotic resistance. There are also 70S ribosomes, which lie freely in the cytoplasm. Bacteria also have a cell wall made of peptidoglycan, also known as murin, and this provides structure and strength for the cell. Mesosomes are also present, which provide energy for the cell through ATP production. And finally, the outer layer of protection is known as the glycocalyx, and this can take form as a slime capsule or a slime layer. Now that we've gone over eukaryotes and prokaryotes, we can now use what we've learned to compare and contrast them. For example, prokaryotes are much smaller than eukaryotes. Prokaryotes do not have a true nucleus, but instead have a nucleoid, a circular loop of DNA lying freely in the cytoplasm, whereas eukaryotes store DNA in the form of chromatin in a nuclear envelope and so has what we call a true nucleus. We also know that what distinguishes eukaryotes from prokaryotes is that prokaryotes have no membrane-bound organelles, whereas eukaryotes do. We also know that the cell wall differs between them, as prokaryotes have a cell wall made of peptidoglycan or murin, but eukaryotes, in plant and fungi only, have cell walls made of cellulose and chitin respectively. We can also compare the cytoplasm as being more dense in prokaryotes and less so in eukaryotes. There's also the fact that prokaryote cells divide or reproduce through binary fission, whereas eukaryotes do so by mitosis and meiosis. Energy production also differs between the two types of organisms, as respiration and ATP production takes place in mesosomes in prokaryotes and mitochondria in eukaryotes. And finally, the size of the ribosomes are smaller, 70S in prokaryotes, and bigger, 80S in eukaryotes. So, in summary, what have we learned? We've learned that a eukaryote is defined as organisms that have a true nucleus, membrane-bound organelles, and examples of these include plant cells and animal cells. We've also asked ourselves, what is a prokaryote, and answered it as being an organism that does not have a true nucleus or membrane-bound organelles, and a common example being bacteria. And then, after comparing the two, we know that they differ in many ways, such as their method of cell division, the types of cell walls, and the size of their ribosomes.